Hello, everyone. Welcome to Rosa's monthly webinar series. My name is Robert Sheehan, and I'm the Director of Modeling at Rosa. We are pleased to continue with our series today on the subject of transforming the discovery of targeted protein degraders, the translational power of predictive PK/PD modeling. Before I introduce our presenter, I would like to remind everyone that our next webinar is scheduled for Wednesday, April 9th. Please keep an eye out on our social media pages and our website for information about that webinar. You can also view any of our past webinars. Just search Rosa & Co., all one word, on YouTube, or go to rosaandco.com for our official website. A recording of today's webinar will be posted in both places later this afternoon. For today's webinar, we're excited to present an early career scientist, Robin Hade. Robin is a modeling and simulation expert in Bayer's Preclinical Modeling and Simulation Group, currently on secondment with Vividian in San Diego. He aims to integrate the experimental data routinely collected in drug discovery to generate a better, more mechanistic understanding that propels projects forward. Robin recently defended his PhD thesis at the Department of Chemistry and Applied Biosciences at ETH Zurich, where he worked on the PK/PD modeling of targeted protein degraders. His seminal papers on the topic have garnered significant attention, and he has been featured as a speaker at several international conferences. At the end of Robin's presentation, we will answer any questions you may have. Please put them into the Q&A by clicking on the button at the bottom of your Zoom screen. And please feel free to add your questions to the Q&A along the way. Robin, thanks for being here and take it away.
Yeah, thank you. Thank you very much for the kind introduction and for giving me this opportunity to present some of our recent work on the PK/PD modeling of targeted protein degraders. As you already said, just a quick recap about myself. My name is Robin, and I just finished my PhD in ETH Zurich in Switzerland. However, as an external PhD student, I was based off campus with Bayer in Berlin. And there, I worked in the preclinical modeling and simulation group, which is why we will be focusing on drug discovery and early development today. So that's it about me. And now let's get right into the topic. And I figured I'd start with something everybody already knows about. So this is thalidomide, a sleeping pill that was marketed to pregnant women in the late 50s and early 60s. And it's quite infamous for causing serious malformations in newborns and was thus soon withdrawn from the market. Still, it's had a great impact on how FDA approves drugs, despite nobody at the time knowing exactly why thalidomide was toxic. Only recently was it then discovered that thalidomide messes with a transcription factor called SAL4. And this discovery, that's really a bit of a game changer, you see, because transcription factors are supposed to be undruggable, like the vast majority of all disease-related proteins, by the way. So if only you could control the power of thalidomide, you might be able to come up with a whole bunch of really cool drugs. And this now neatly brings us to targeted protein degraders, where you take thalidomide, that's this blue triangle now, you put it on a leash, and you link that to a targeting warhead, which is depicted in red here. And this whole thing, that's the degrader. The red moiety then binds to the protein of interest, POI. So this abbreviation is going to be important. POI, that's the one causing the disease. While thalidomide binds to an enzyme called an E3 ligase. And this E3 ligase, in turn, mediates the ubiquitination of the target protein, and it does so over and over again. Thus marked, the protein is then recognized by the proteasome, where it's finally degraded. And you can think of this like paper in a shredder. And what makes this whole process targeted is this red warhead moiety that directs thalidomide to only degrade the intended target protein instead of running wild like it did in the 60s. Okay, so now that we know what we are dealing with here, let's get modeling. And the first thing we do is we take the cartoon from the previous slide and analyze it through the lens of the 4 pillars. That's a conceptual framework where you break down a drug's mechanism of action into a sequence of 4 critical steps. And this divide and conquer way of thinking makes it easier to wrap your head around what's going on pharmacology-wise. So in order for a degrader to elicit a pharmacological response, it first has to get into the target cells, where it must then bind to the target protein itself, as well as to an E3 ligase to form a ternary complex. Ternary because it's made of 3 constituents. In this ternary complex, the E3 ligase then mediates the polyubiquitination of the target protein, which is subsequently degraded by the proteasome. And finally, this degradation translates to a downstream pharmacodynamic response, so you get some sort of biomarker readout. At least, hopefully, you get some sort of biomarker readout. Great. But what's all that good for? Now, we've identified 3 key areas where translational PK/PD modeling can really propel projects forward. And the first one of those is about lead optimization. So the goal here is to increase the potency of your degrader by tweaking its binding affinities. So what we have here is the levels of the POI, the target protein, right? So that's the one we want to get rid of, plotted against the degrader concentration, and all of that's in vitro. As you can see, this compound A, so the black one, does degrade that protein. However, at high concentrations, there's this weird behavior of the graph going up again. Now, this phenomenon is called the hook effect in reference to the shape of the graph. So it looks like a hook. And what's going on here is that we need this ternary complex to get degradation. But if there is too much degrader around, then each E3 ligase and each target protein, they'll have a degrader molecule all for themselves. And therefore, they won't have to share a common one between them because there's just too much around. And thus, you get less ternary complex and therefore less degradation. Okay, now, speaking of less degradation, now this compound A looks rather miserable now that I think of it. Everybody in the team would be way happier if instead it was more like this red compound over here that's much more potent. To get there, you first need to determine the binding affinities for compound A to understand why it is so bad. You then feed that information to the model before fitting it to the experimental concentration response profile as shown in this plot. And now the model allows you to make an a priori prediction about how much the different binding affinities will need to be improved to achieve the red profile. So the affinity for the protein of interest would need to be increased by a factor of 10, the affinity for the E3 ligase by a factor of 5, and the cooperativity by a factor of 1.5. And as it turns out, medicinal chemists have been able to synthesize such a compound, so a compound with those improved binding affinities, which was then run in this degradation assay. And as you can see, potency really was improved to the extent predicted, which validates the model. So that's very nice. All right, now that's all neat and dandy. But in vitro assays, they can only ever tell us that much. So let's see how those targeted protein degrader drugs, how they perform in vivo. If only those pesky PK/PD studies wouldn't take so darn long. Now, instead of waiting for weeks on end until study results come in, you can also take advantage of the data you already have. And the way this works is you take your run-of-the-mill animal exposure study, where you measure drug concentrations in plasma, and you combine that with in vitro data on potency. It's what we were just optimizing. Now, what you get out of that exercise is the full time course of protein degradation for different doses of your degrader. And while you're at it, you might as well incorporate data on the expected experimental variability to come up with those shaded prediction intervals. Let's now have a closer look at that output plot. Now, as you can see, the model unambiguously predicts that you will indeed see degradation for all 3 doses, but only a little bit for the lowest one. Next, you can predict that it will take 5 hours until you see the first degradation. then the maximum extent of degradation should be observed after 2 days, and detectable levels of degradation should persist for a week or so. Okay, now let's have a look at the actual experimental data to see how the model did. Well, that's not too bad, is it? Let's take a moment to reflect on what just happened. So we already had the predictions. So that's the simulated lines and the shaded 90% prediction intervals before the study was even performed. Then we saw that the experimental results confirmed the a priori prediction. Thus, we've just established that modeling can answer these key questions that commonly arise when planning studies, right? So these key questions here. But I mean, what's in it for drug discovery projects? Now, to find out, we'll compare the actual study protocol, which was designed without any regard for modeling, to what the model would have suggested. And let's focus on the measurement time points for now. So for the first three, right? So those are the ones the model suggests, right? The ones above the x-axis. Those are the ones where the measurements have actually been performed, right? Now, for the first three of these measurements, I think there's not really much of a difference. However, they also did a 2 weeks time point, they being the authors of the study, where there's absolutely no degradation, just as the model predicts. This measurement, therefore, only adds very little to the study and could have easily been avoided by performing PK/PD modeling, thus reducing the number of animals in the study. Yes, the model-informed protocol that would have also saved valuable time. Now, in a drug discovery setting, of course, you don't just have one degrader that you're interested in, but usually you have a whole bunch. For the sake of this presentation, though, let's start small with just 2. On the left-hand side, we have the compound from before. While on the right-hand side, we see a new one. So first, let's do the whole study planning routine again, which would have saved plenty of more rats. But wait a bit. This compound 6 here, so the new one, that's predicted to be much better. That is, requires a lower dose to achieve degradation than compound 4. And the experimental data also confirmed this prediction. Therefore, you could have skipped studying compound 4 altogether, instead focusing on the far more promising compound 6 right away. Or in other words, modeling can help you find the best degraders. So now you know which compound's best at degrading. There's just one tiny problem, and that is that no one in discovery has the slightest idea how much degradation you actually need. However, the clinicians, they can often tell you how much of a certain biomarker response they are aiming for. In the example that we've been studying so far, the protein of interest, POI, so that's the bad protein, the one you want to get rid of, so the one that's getting degraded, that's called RIPK2. And the effect you want to get is reduction in pro-inflammatory cytokines, such as TNF-alpha. Now, for this slide, the cells were incubated in the presence of various concentrations of degrader, and both the extent of degradation and the downstream effect were determined and then plotted against each other. Right, so the y-axis here, that's about TNF-alpha response. So not protein degradation, but the downstream pharmacodynamic readout. While the x-axis, that gives the corresponding protein degradation rather than drug concentration, right? So 13% RIPK2 degradation, RIPK2 is the target protein, 13% RIPK2 degradation already causes 50% reduction in the TNF-alpha response, which is quite impressive. Okay, now, of course, what you really care about is the vivo setting. So let's pick up where we left off earlier, predicting RIPK2, so target protein, degradation for the good compound. And now you take those predictions and you feed them into the translational model from before. And that model then converts the predicted degradation into the PD response. So finally, you get a prediction for TNF-alpha release, right? So here we have TNF-alpha release, so the downstream pharmacodynamic response. Here we have the protein degradation, protein levels remaining, and both of those are predictions. So the only experimental data where a model was fitted to is shown here. So that's the in vitro data that's used to connect protein degradation to the downstream pharmacodynamic response. Okay. Yeah, so we're back here, and as you can see, so this model prediction works pretty well. So the experimental data confirm this a priori prediction. And next, let's compare the two plots, because what is a bit surprising is that while there is hardly any degradation for the blue dose, there's quite a strong effect on the PD response, roughly 80%. And that might already be enough. Right, so as you can see here, blue dose gives very little degradation, but it gives quite a strong effect on the pharmacodynamic response. So why is that? And just this observation that there's this disconnect, that's really not a trivial result, as these plots now illustrate. The one to the right is what we looked at before, just for a single dose. While the one to the left shows the experimental PK profile. And I've also taken the liberty of indicating the functional potency from a whole blood assay, right? So this IC50 value, that's already corrected for plasma protein binding. Now, if you were to just eyeball the raw data here, you might conclude that inhibition should persist for no more than a couple of hours, because that's where you have the IC50 covered. But actually, it's several days that you have inhibition for. Now, this goes to show that the IC50 value on its own doesn't tell you an awful lot, and it's only through modeling that you can properly interpret the data. Yes, okay. Bobby's seeing the question, so that's very good. Right. Properly interpret the data. Now, splendid. But where's the clinical relevance in all of this, you might ask? Okay, so here's the deal. What we've been discussing so far, that's just step one. And the goal here is to build confidence in the model and the assays used to inform the parameter. And that already works pretty well, right? So what we do is we validate the model and the assays that inform the parameters, which are, of course, out of the model. And then we do that with animal studies, right? Now, to unlock the full potential of translational PK/PD modeling, you simply replace the animal parameters with human parameters. That way, you can account for species differences in the pharmacology, because remember, you want to treat humans rather than rats. Now, once you have done this replacement, you can assess the merits of different. You can assess the merits of different compounds based on their projected dose in patients, right? And of course, what you need to do, a very important part of what you do here, is you have to predict the PK in human. In the animals, you could just measure the PK. In humans, you have to predict that using PBPK modeling or allometry or the like. So this concludes the first part of my presentation, where we discussed how modeling can guide medicinal chemistry efforts. We established that modeling can help you figure out what studies to perform on which compounds. finally, we saw how seemingly nonsensical data can only be interpreted through modeling. And now with so that's the general idea. And now I want okay. So that was the general idea, and we used literature data to illustrate that, right? However, we also used this model for several in-house projects. And what I'd like to do now is to go through a case study to explain and show how we used these modeling techniques in this particular example. Now, I'm not sure what the plan is. Maybe Bobby can tell me. But perhaps it would make sense if there are questions about the general idea to address those now and then do the case study, or I can just proceed with the case study and we do all of the questions at the end. But maybe it makes sense just because if people don't get like the general idea, then I'm not sure how much use the case study will be.
Well, we can pause for a couple of brief questions if you'd like. I certainly want to be mindful of time to make sure you can finish your full presentation as well. But I agree, it might be helpful to just touch on a couple of key questions here.
Or maybe everything's clear already, so then I mean we don't need to stop, but just in case.
Yeah, I think a couple of these might be helpful overall questions to really capture. So we'll just do a couple of quick ones for a quick breather, and then we'll dive back in. One question is, how often do you actually see the hook effect in vivo?
So I'll just go back now to the slide. Yeah, maybe it's nice this way. It's a bit more interactive as well. So this slide. So I think that's the one where I talked about the hook effect. So I personally, I don't think I've ever seen the hook effect in vivo. We just published a paper where we stated that I think the phrasing we used was that the hook effect doesn't need to be at the forefront of researchers' minds when studying degraders in vivo or something along those lines. So what we're trying to get at in very diplomatic terms is to say that it's not so important in vivo. In vitro, we see it all the time, really. And so I think it makes sense if you're going to use the in vitro data and you know that this hook effect, that's not like just because the assay failed, because the cells died, because something went wrong, but that is like an intrinsic part of the mechanism of action. I think it makes sense to include that in the modeling, to use all the data you have rather than just, I mean, the alternative approach would be to just ignore like these data points. But I don't. So I think that's not really good scientific practice. So about the hook effect, in vitro, very important, shows up all the time. If you want to translate in vitro data to in vivo, then I think it makes sense to include it in the model so that you're sure that you don't miss out. In vivo, I think I might have seen it once. So there's one example where where you reach we reached the necessary concentrations, and then we saw like a mild reduction in degradation in line with what you would have expected, but it's very minor, and so it's not really a massive deal. Yeah, and I think one of the reviewers for the latest paper, so he said, or they said, that they saw the hook effect in vivo in unpublished data. I don't know. But yeah. So let's just take that on face value.
That makes sense. Next question. So you showed PKPD data looking at concentrations in the plasma that you used for your modeling. Do you also account for distribution of the compound to the site of action where the POA degradation is desired?
So now this is going to be part of the case study that I'm going to present momentarily. in general, when we do the predictions at this stage, right? So the stage is lead optimization, roughly, where we have a lot of compounds. We don't do so we just have the plasma concentration data, and then we assume that it's the same. Only if that fails, if the predictions are off, do we start to think about why that might be, and then we would look into distribution of the drug to the site of action as one, as you'll see in the case study section, is one important mechanism. I think you're on mute.
Thank you. I think I'll ask just one more question before we move on, because I have two kind of related questions. They also may relate to something you'll touch on in your specific case study. But in general, is the turnover rate and induced degradation, is that going to be kind of similar in all cases, depending, or is it going to be dependent on the POI and the specific complex? And if it does vary, how much does the turnover rate matter in predicting the downstream effects?
So if I get the question right, then it's about whether the protein turnover is different for different proteins. So that's like the first part, and then whether those differences matter. So it is different, obviously. So I think there was one paper where they found the median protein half-life to be 48 hours. So when you have no idea, then I think going for 48 hours is an educated guess. We always use so we never use this default value actually, but we always go with experimental data. You can get those from SciLec experiments, for example. There's a lot of published literature on protein half-lives in different tissues, different cell types and different tissues. There's both data on protein half-life in vitro, and then there's also data on protein half-life in vivo, so in mice, I think, where they look again at different tissues, different proteins. So there is good data on that. The data does vary quite a lot between different cell types, especially if you're looking at immunology. You have the fast turnover cells, such as monocytes, which often have shorter protein half-lives. And then you have slow turnover cells, T cells, for example, so plasma cells, which have longer protein half-lives, and those can vary by an order of magnitude. So that's the differences, and then of course if you compare different cell types, then that might also be different. One thing that I've also been thinking about recently, but that's not in this presentation, is how cell turnover, so not protein turnover, but the entire cell being turned over, also being replaced in vivo, of course, how that affects protein half-life and the recovery of protein levels in vivo. Right. And why it matters. So we can just go to this slide maybe. So the protein half-life parameter, what that does is it tells you where this nadir, so the lowest point of the curve, where that is found, so at which time point, and then also how long effects persist, and that's important. Not so much if you're going for repeated administration. So when you're doing daily dosing, for example, then protein half-life doesn't matter an awful lot for the drug effects at steady state. However, if you're doing single dose administration, such as you always do in discovery and early development, right? When you're testing a drug for the first time, you just give a single dose, and then you need this parameter to understand the data, right? To make sure that what you see is actually actually make sense in the context of what you already know. So that's why it matters. Again, it doesn't really matter if you're doing daily administration and then only looking at protein degradation at steady state. Okay.
Great. Thanks. That makes a lot of sense. We're getting a couple more questions, but I would recommend going into the second half of the presentation since we're
Yes.
approaching
So
the
the
halfway
second half
point.
is much shorter. It
Right.
should be fine. Should be fine. Okay. So now, yeah, now it's always with the animations, right? So if you have to quickly go through the slides, then the animations mess everything. Okay. So let's just go back here. So that was the general idea, right? Illustrated through literature data. But we also use these models for several in-house projects at Bayer. And for this particular one, so the one I'm going to talk about, the team had already identified several lead compounds when we first got involved, and they had run the usual degradation assays on those, and they had also assessed exposure in mice. So that's the data we already have. Now, they wanted to set up the first PK/PD studies, which is when I got modeling. And my aim here was to help the team find the best compounds and dose schedules to show strong degradation in the xenograft mice, right? So this is an oncology project. So we're talking about xenograft mice. Right, so the way I would do that is by leveraging the in vitro degradation data for a priori predictions, just as I've outlined before. And again, since this project was at such an early stage, we would have to do that for a whole bunch of compounds. We hadn't decided for a specific drug just yet. Now, as you will be all well aware of, things don't always go perfectly smoothly in lead optimization. And therefore, I want to talk about some of the hurdles that we had to overcome and how we managed to do so in this second half of the presentation. And yeah, so here, just as a quick reminder, what we do is we take the in vivo PK and the in vitro PD data to predict protein degradation in the tumor-bearing mice, right? So, but what's that? I mean, there's two graphs in that plot on the right-hand side. Now that's because we had two different unbound in vitro DC50 values from two different assays using two different culture media. And while those should, in theory, give the same result, in practice, they very much did not. Now, we decided to resolve this dilemma by performing an in vivo PK/PD study, which is shown here, which is designed in such a way that we could clearly tell which of the two assays was right. And lo and behold, turns out the blue one, so the blue assay, that's pretty much spot on, while the red one is rather far off, right? So now we know that when we want to make predictions for the next compound, we should go with the 10% BSA assay. So that's this blue one, rather than the red one. And yeah, and figuring out which degradation assay is relevant isn't only interesting to us as modelers, but also to the chemists when they are building their structure-activity relationships for optimizing potency. Now, moving on to the next compound, where we also encountered the next problem. Because you see, the degradation assay from the previous slide, that actually only gives us the DC50, so the degradation potency, in this 10% BSA media. So that was the incubation media for that assay, which we then still need to correct for non-specific binding. Because plasma protein binding in vivo is different than in this artificial media, right? However, that parameter, so the fraction unbounded media, that hadn't been determined for this drug just yet. Thus, we decided to predict it with a separate model and then use that value to, in turn, predict degradation in vivo. Okay. And once again, the project team was very happy with our work. Yeah, and later that FU value that could also be confirmed experimentally. Now, this second model for FU prediction, so that's not related to like the main part of the presentation, but that's a side quest, if you will. But this second model for FU prediction working so nicely, that's good news, because it means we no longer have to wait for the fraction unbound immediate to be measured, which always took quite a while, right? So instead, we can use a predicted parameter to in turn predict protein degradation. Okay, so now let's shift gears with the next compound. And here, I want to illustrate that translational PK/PD modeling can still be useful even after the results of the actual animal studies have already come in. I mean, just look at those data. I mean, they're already all over the place, which unfortunately is quite common for in vivo degradation, right? Which I want to draw your attention here. As you can see, there's only 2 data points at this time point, which is because the third one, so one additional value, was above 140%. And yeah, the upper limit for the y-axis is 140%, so this value isn't shown. Just keep in mind that there's like additional values somewhere up here. Right, so data very messy, which unfortunately is quite common for in vivo degradation. Anyway, we can still fit a PK/PD model to get this. Now, what we do here is we use an empirical model. That is, we use the data that's shown, so the black data points. So those now come first, right? And then we come up with a line that kind of goes where the data points are. So empirical fitting to the experimental PK/PD data. Now, this graph already looks much nicer than the data. The only problem is that the confidence intervals are absolutely massive, right? So not even the model can really make sense of the data because they're just too messy. But you know what data aren't messy at all? The ones from the in vitro assays. And wouldn't you know it, our a priori predictions allow for a confident assessment that's not affected by the uncertainty associated with the in vivo data, right? So here, our predictions are even more informative than the actual PK/PD study. At this point, I just want to stress the difference between the blue and the red once again. So the red graph, that's based on an empirical model. Empirical in the sense that the model parameters, as shown here, are informed from the PK/PD data. So here, data comes first, then we get a model from those data. Data being the PK/PD, so the animal efficacy data. With blue line, that is independent of the data points shown here, right? Because that is informed from orthogonal assays, so from in vitro degradation assays, among others. And because those assays, you can perform them at a very high volume. So this example, the degradation assay would be performed on every new batch of compound as a part of the quality control. So we had an enormous amount of data. Therefore, you have very high precision for your parameters. And so you're very, very confident in the parameter values. Whereas with the in vivo data, so the red approach, the in vivo variability screws up your estimates, unfortunately. Okay, so blue prediction line comes first, data confirm line. Red fitting empirical, data come first, and then the line follows the data. Okay. Let's stick with our predictions then, right? Now, at this point, I should probably confess that I haven't been perfectly truthful so far. You see, what I've shown you up here in the PK plot, those were always the profiles observed in the PK/PD studies themselves. However, for our predictions, those aren't available yet. Instead, we use a dedicated exposure study to inform the model. And that approach resulted in the following prediction, which looked fine. So the study was performed, and ouch. I mean, everybody in the team was really happy that this compound outperformed our expectations, but we modelers, we weren't quite content with the prediction. Fortunately, our colleagues had also measured drug concentrations in this very PK/PD study. And yeah, look at that. They are in a completely different ballpark. So I should stress that these two studies should give the same result, right? Because you're just dosing the same drug to mice, several mice each time. Once you get very high exposure, once you get very low exposure, for no good reason. Now, if we use the blue profile as input to our model, we get different prediction, if you can call it that, which is way more accurate. So what do we make of this? Well, experimental irreproducibility is a kind of kryptonite for all predictive approaches, really. And I'm afraid that is a limitation that we have to be aware of and that we have to cope with. Okay, but let's not dwell on such gloomy thoughts any longer. Here we have another prediction, and oh no, not again. And this time we can't even blame it on the exposure study. But wait a minute. The red graph uses plasma PK as an input. Plasma. But this is an oncology project. So shouldn't we actually be looking at tumor exposure instead? And so now this brings us to the question that was asked earlier. So I'm glad we can now answer that. So plasma is what we used, but this is oncology. So what does the tumor profile look like? We see the distribution is actually quite slow, right? So it takes some time for the drug to get into the tumor. So what happens if we use the tumor PK as an input to our model? That's much better. Now here we've informed the fraction unbound in tumor from the in vivo degradation profile, which results in a nice fit, and the estimated value also makes sense mechanistically. Now, unfortunately, we cannot use the tumor PK for our predictions, as those need to be done before the data from the xenograft animals are available. We will, however, continue using the blue model when planning studies on this particular compound. Right? We can't use them for predictions because predictions are done using the exposure in non-xenografts, in normal mice. They don't have a tumor, therefore we don't have tumor PK. But once we did the first tumor studies, we know about the distribution behavior into the tumor, so we can incorporate that to the model. Okay, so what was our modeling in this particular project good for? And I hope I removed all of the references to what the project's called. Okay, so what was it good for? First of all, modeling allowed us to identify the relevant in vitro degradation assay. Next, modeling sped up drug discoveries. We don't have to wait for assay results to come in. Also, it helped make sense of the erratic in vivo degradation data. Then it highlighted the need to further optimize the exposure studies to make them more meaningful. And finally, modeling revealed the tumor penetration for a particularly interesting compound was slow, which might be relevant for translation into the clinic. Okay, so now I would like to thank my Bayer supervisor, Andreas Reichel, for his mentorship and his invaluable scientific input. Furthermore, it would be remiss of me not to mention at least some of the other great colleagues that I've had the pleasure of working with on this project and on others as well, and who greatly inspired my modeling and who generated all the data that I just showed and used for illustration. A huge thank you also goes A huge thank you also goes to my supervisor at ETH Zurich, so that's Professor Kremer, for enabling me to conduct my PhD in this fashion, and for her unwavering support throughout. And finally, I'd like to give a shout out to Pharmetrics grad school that I attended, where they have great courses for people doing their PhD in the field, and they also get you in contact with industry. So that's a shameless plug for any PhD students or master students perhaps watching. Okay, speaking of shameless plugs, with that, I'm done with my presentation. Thank you very much for your attention, and I'm now looking forward to an interesting discussion and questions as they relate to either part of the presentation. Thank you very much.
And thank you so much, Robin, for the wonderful presentation. As you said, from now we can get back into some questions. And again, audience, feel free to keep adding more to the Q&A. First, we have a question. So you talked a lot about translating between rodent and human models. When you're doing that, do both the protein turnover and the induced degradation rates need to be scaled, or can those be assumed to be similar between rodent and human?
Yeah. So, so the induced degradation rate, that doesn't get scaled. So I don't see, so not at all. With regards to protein turnover, I'm not really sure whether that scales. So it seems to me that just on like so if you're talking about the order of magnitude, right? Those are pretty much the same between human and animals. I think that makes some sense because the cells are, I mean, the cells are the same size. So what scales are physiological parameters on the macro scale, right? So blood flow scales. But that's because right, but that's like because the vessels have different sizes and the ratios between surfaces and volumes change. But the individual cells, I mean, and a mouse cell is just as large as a human cell, like for the most part. And therefore, I think it makes sense that they don't seem to scale. So protein turnover rates don't seem to scale, at least not like with regards to orders of magnitude. So the answer to your questions, for like all I know, none of the two parameters scale.
Thank you. Okay, next. Which PPB method has been used to measure the unbound fraction in plasma and tissue? If you're
So
aware of the experimental.
yeah. So the thing, so that's in-house data, right? And we have to go through a clearance of material that we present or publish. And the information on which method was used here, I think that hasn't gone through. Sorry, I know that hasn't gone through this clearance process, so I can't really answer that question. What I can say is just that fraction unbound for proteins is often very low, so it's important to optimize that method, and there are several approaches. There's one that's equilibrium dialysis, the standard method, but then you also have ultracentrifugation, ultrafiltration. I think that's also one. And then there's a transseal assay. And I can also comment that the colleagues, so that would be Stefanie Zimmermann at Bayer in particular, they spend quite a lot of time optimizing their methods. However, I'm afraid I think I mustn't talk about the technical details here. But that's irrelevant. So that's important, I know. If you don't have the value, then what you can always fall back to is predict the relative fraction unbound. So actually, you don't need the fraction unbound in the media and in plasma. You just need the ratio between those. And since the dilution ratio, so 10% BSA is like 10% plasma, so that ratio is 1 to 10. You can use this dilution method to predict that the ratio between the fraction unbound values, that should also be 1 to 10. That's very rough, And then you can go into, again, technical details for how to improve that prediction for diffraction. But I think a reasonable starting point is to go with this 1 to 10.
Yeah, that makes sense. Great. All right. Staying on some of those in vitro assays, so you discussed that scenario where you were getting very different outcomes from your different in vitro assays, and you were able to kind of pin down which one was more reflective of the in vivo data. But were you able to, or could you envision backing out exactly what caused the discrepancy between the two assays, and could you use that to inform future assays? Is there a better use case for one or the other, either compound specific or indication specific?
Yeah. So this is very hard to do because I mean the people who set up the assays they didn't intend it to be wrong, right? So it's like it's not the idea. So and I'm not an experimentalist. Again, this is very hard. So like doing the experiments is very hard. I fully acknowledge that, and I'm not an expert on that. But what I will comment on is that this 10% BSA medium, that's kind of the standard assay. So that's the default. Whereas this one that uses basically human plasma as a medium, that's more niche, let's say. And so for whatever reasons, so I think that you can come up with several reasons why this specialized assay that would also require further fine-tuning of, I don't know, the analytical methods or so that's just non-standard, right? And so if you're doing something non-standard, then you encounter initial hurdles that you have to overcome. And my interpretation would be that those haven't been overcome yet. Now that is a very, very, very surface level interpretation. But again, I can't really talk about the technical details, so the experimental technical details. First of all, because I'm not an experimentalist, and second of all, because that hasn't undergone clearance.
Understandable. Thanks. All right. So you've also discussed how this kind of modeling can give kind of real world benefit by reducing the size and duration of some of these animal trials and reducing the number of animals you're using. From a practical point of view, have you faced difficulty getting buy-in on that idea? Do you think there's enough confidence in the model from all the parties to actually go forward with having smaller scale trials based on the modeling predictions?
So we don't necessarily need to have smaller scale trials because I mean, so this is like so you saw the case study, right? So it's still you have like 5 time points. What we are doing is we are helping with compound selection. So okay, so like back up a bit. Without modeling the way so the studies are still designed, right? So people don't just do random studies, but they do look at the data and they do have a rationale for how they come up with the number of animals, the doses, and the measurement time points, and the compounds they want to use. The modeling does the same thing, right? It uses the same inputs, but it does so in a internally consistent manner. So it kind of does the maths right instead of just eyeballing and making assumptions and so on. So therefore, it's more objective. So that's one huge benefit. So instead of project teams going back and forth between slides with raw data and kind of just voicing their opinion, you can do these simulations. Then everybody looks at the simulation, right? We agree on what input assays we want to use. And then that's the basis for study planning. It's also useful for communicating with regulatory authorities because they often want to see power calculations for why you choose to go with a certain number of replicates. And you can do those on a sound basis, right? So you can say, okay, we work with, I don't know, 3 animals in the control group because that will give us, and then you can come up with some power, so statistical significance arguments. So that's that. Yeah, and then of course it's still you still do the magical time points, right? So you never do for example, 7 hours, because 7 is a nasty number. So 6, very nice number. 24, extremely nice number. 2, also quite nice. 3 is sometimes okay. 1, almost never. So that's kind of, right, so those metaphysical factors also play too. Right. So what was the question? I got a bit lost.
So I think that was just do you have trouble getting buy-in on the confidence of these predictions to actually use them to make
No,
make
no,
your
no, that's fine. No,
experimentation
no,
more
again,
effective?
that's fine because you have to plan the studies anyway.
Right.
And I think everybody agrees that this is like the more stringent
Yeah.
way.
Great. Great. All right. So do you think the DC50, Dmax, and half-life parameters are sufficient to predict the in vivo degradation kinetics, or do you also need to consider the kinetics of binding, so a more complicated KCAT model with more parameters that might be harder to get in the earlier stages of discovery?
So when you say the KCAT model, so that is a term that I came up with. Um, yeah. So the beauty in all of this, and I'm very proud of that because it's a lot of maths work behind it, which nobody cares about in the slightest. But the thing is that, so what we call the KCAT model and what we call the extended hook model, which is the models used in the slides shown here, those are the same. They just use different parameters. So the way you can think of it is that the KCAT model and the model used in vivo. So the KCAT model is the model we used for the in vitro. So the first use case. And then for the second use case, we use this other model, the in vivo model. And those are the same in the same sense that describing a PK profile, either using clearance and volume of distribution, that's the one, or using a different parameterization, such as, I don't know, the clearance and terminal elimination half-life, or the micro constant, right? You could also use the distribution between different compartments that can also be described just with rate constants rather than flows. And so those are basically the same model. They just use different parameterizations. And in the same sense, the models we use here are the same model, just using different parameterizations. So you can calculate DC50 from the KCAT model parameters. Right. And so for that reason, no, there's absolutely no reason why you would ever need to use this more complex model if you're talking about like our KCAT model. Now, when you're talking about the binding rate constants, those are not in the KCAT, so what we call the KCAT model, because the assumption there is that binding is much faster than degradation. I think that is quite reasonable. But if you know that it's not the case, then you might have to do that. But then again, so you have to do the surface plasma resonance or whatever studies to determine the binding rate constants. I think in most cases, your time and effort could be spent more productively. That's just my opinion.
Understandable. Okay, well, I do think we are starting to run short on time. So I think I'll just thank you for your time and for the wonderful presentation, Robin, and wish everyone a good rest of your day. And we hope to see you again at the next Rosa webinar.
Okay, my pleasure. Thanks, Bobby.
Thank you.